Okay, good morning. Let's get started. I think a couple of students, they probably registered a little bit later, so they were kind of still looking for the partners, group member. So if any of you does not have a partner or group, please email me. All right, so I'm trying to match you guys, right? And also I updated the class notes for carbohydrate metabolism 1 and 2. I'm sorry, I realized a couple slides actually are missing. So I sent you guys email and also updated Canvas, right? And today we're going to finish the digestion, absorption, lipids, and also we'll talk part of the carbohydrate metabolism, right? And we also have informal writing, right? Assignment. Okay, so I think I already talked about it. The lipids absorption, right? I think I will finish this slide. So try to remember the structure of the triglyceride. It's very abundant in our food, in diet. And also, cholesterol, phospholipids, and free cholesterol, cholesterol esters, and also talk about the concept of free versus esterified lipids, right? So esterified lipids, basically the fatty acids attached to the other component of lipids, right? For example, triglyceride is considered phospholipids because 3 fatty acids molecules attached to the glycerol backbone, right? And also cholesterol esters is a combination of 1 molecule of free cholesterol and 1 molecule free fatty acids, right? And fatty acids will consider the free lipids, the free fatty acids, or just free cholesterol molecules, right? It's also free lipid. Okay, so digestion of lipids. So where does lipids digestion occur? Again, we know the most abundant lipids in our diet is a triglyceride, right? So triglyceride digestion begins in the stomach, and so about 10 to 25% of triglycerides digested in the stomach by the enzyme called gastric lipase. And of course, the small intestine is the most important site, right, for digestion for lipids. So the rest of, the triglyceride or lipids is digested in the small intestine and by a different enzyme called a pancreatic lipase. So that means the lipase that digests lipids in the small intestine is coming from pancreas, right? The pancreas release the lipase. And gastric lipase usually digests the SN3 position of triglyceride. That means they remove fatty acids from SN position of glycerol backbone, right? And generating a 1,2-diacylglycerol and 1 free fatty acid molecules, right? So gastric lipase usually remove the 1 fatty acid molecules from SN3 position of a glycerol backbone and release 1 free fatty acid molecules, and the other one is diacylglycerol, means the glycerol backbone still has the 2 fatty acids attached, right, to the SN1 and SN2 position. And pancreatic lipase, that's a little bit different things, and they actually cleave the 2 fatty acids from glycerol backbone at SN1 and 3 position, right, and release releasing one monoacylglycerol and two free fatty acid molecules. Okay. And in small intestines, there are also other type of enzyme that can digest in other type of lipids. For example, phospholipase A and B. These two enzymes are also secreted by pancreas. And hydrolyze the phospholipids basically also removes fatty acids, right, from phospholipids. Remember phospholipids molecule structure, right? It has two fatty acids attached to SN1 and 2 position, and SN3 position basically is linked by a phosphorylcholine group, right? And also cholesterol esterase secreted from pancreas and hydrolyze cholesterol esters, right? Remove fatty acids from cholesterol esters, right? And generating one free fatty acids and free cholesterol molecules, right? Okay. Now, so digestion is kind of simple, right? Just one, mainly, for triglycerides, just the lipase, right, to remove the fatty acids from the esterified lipids. And now we talk about absorption. So this again is the enterocyte. Enterocyte is most important type of cells in small intestine, and that's the absorption function. And if you still remember, we talk about the water layer, right, above the brush border, and this water layer somehow protect absorption of lipids, because lipids is not water soluble. So that means our body needs to do something to help lipids absorb into the cells and pass through the water layer. So there is some structures called vesicles, bile salt vesicles, and also called meso, right? This vesicles basically, it's just formed by a lipids molecules, right, and together with the bile acid. So bile acid help to restructure the lipids to make this hydrophobic portion of the lipids inside of the vesicles and expose the hydrophilic portion of the lipids outside, right, the vesicles. So these vesicles were able to efficiently penetrate the water layer, so it can more efficiently, absorbed by the enterocytes, right? So again, bile acid or bile salt vesicles is a way that facilitate lipid absorption. So again, the bile acid is very important, right? One of the function of bile acid is to facilitate the lipid absorption. So if someone has some problem with, the secretion of the bile acid, then that probably will have some problem with the lipid absorption, right? Any questions so far? Okay, so once lipids, like basically free fatty acids, right? Once free fatty acids or cholesterol is absorbed or transported into the enterocytes by bile salt vesicles, there are actually additional steps that occur in the enterocytes. So there is type of a protein called fatty acids binding proteins, or the lipid binding protein, depending on what type of lipids, right? It binds fatty acids called fatty acid binding protein. It binds to the cholesterol binding protein. So the let's say here the fatty acids binding proteins. Once fatty acids is entering the enterocyte, it binds to the fatty acid binding protein. Remember, the lipids is never be able to, like a molecule present as a free molecules. Usually need to bind protein to help lipids transport, right? Either in the circulation or inside of cells. So fatty acids, once is absorbed into the enterocyte and binds to fatty acids and fatty acid binding proteins, and then transport fatty acids to the ER component for the lipid synthesis or other process of transport. So that way will make a free fatty acids concentration low, right, inside of the cells, and generate sort of the gradient. So you have a high concentration of fatty acids outside the cells in the lumen side, right, and a lower concentration of free fatty acids inside of cells. So that will promote more fatty acids being transported into the cells. That makes sense, right? You generated sort of a gradient, like concentration gradient. You have high concentration outside the cell, low inside of cell. So that will promote more fatty acids being absorbed into the cells. So this is sort of the mechanism. I think I will talk about this, right? Fatty acid binding protein and also sterol carrier protein that transport cholesterol to the ER. And once the free fatty acids being transported to the ER, then free fatty acids will have to be re-esterified to triglyceride, right? Re-esterify means that free fatty acids will attach to the glycerol and generate triglyceride, right? This process, the re-esterification process. So triglycerides, so free fatty acids enter the cells and re-esterify to triglyceride, and triglyceride cannot be simply transported directly to the circulation, right? It has to be packed into the called lipoprotein. Have you heard about this, right, before? So lipoprotein is a group of protein that help transport triglyceride from one tissue organ to the another tissue organ. So in the small intestine, the enterocytes usually can synthesize the one type of lipoproteins. Do you guys still remember what type of lipoprotein that can be synthesized in the small intestine? Like chylomicron, very low density lipoprotein, or high density lipoprotein. Which one? Do you remember? No? Okay. Chylomicron. Okay. Chylomicron is one of the lipoproteins. So it can be synthesized in the enterocytes. So the purpose of this, that can transport a triglyceride that initially absorbed from the intestine, right? And transported triglyceride from small intestine to the adipose tissue for the storage, right? But we will talk about this later, right? Specifically, or more detail. But just, in general, it's called the general steps, how lipids being digested, absorbed, and transported, right, to the destination. And also the other concept that you need to know that lipids or triglyceride is not directly transported into the circulation, but it's transported to the lymphatic system first, and then to the circulation. All right. Any questions? Right. Yeah.
When you say TAGs, is that just like triacylglycerol, or what is TAGs?
Oh, TAG, triacylglycerol. Yeah, sorry. TAG, tags, right. And diacylglycerol, DAG, DAG. Right. Okay, yeah, I think we talked about this already, right? So chylomicron. Okay, so this diagram gives us probably a little bit more details that help you understand the transport or absorption of the fatty acids. Let's say the dietary triglyceride, right, digested by lipase in both stomach, only 10 to 25%, and the rest of triglyceride is digested by pancreatic lipase, right, in the small intestine. And the digestion basically means that break down the triglyceride molecules and release free fatty acids from glycerol backbone, right? And basically after digestion by lipase, will generate the two products. One is the free fatty acids molecules, right? And the other one is the monoacylglycerol. That means you still have the one fatty acids attached to the SN2 position of glycerol backbone. This represents glycerol backbone, right? And these fatty acids can be absorbed by the enterocyte, and the process, this absorption process requires the bile salt vesicles, right? Remember this. So the bile salt vesicles facilitate this absorption process. Once enter the enterocytes, the free fatty acids can bind transporters fatty acids binding proteins, or also called transporter, right? From the absorption site into the ER, and to re-acidify to the triglyceride. And this triglyceride basically is packed into the chylomicron, which is one of the type of lipoprotein, right? And chylomicron then carry the triglyceride from enterocytes first of all to the lymphatic system, and then eventually to the portal vein, right? And majority of the triglyceride actually being transported to the fat tissue, adipose tissue, and stored in the adipose tissue. Okay, so I think now we finished the digestion, the absorption part, 3 macronutrients, right? And if you still remember, there are actually a slight difference in the digestion absorption process between the macronutrients. So, for example, carbohydrates and lipids digestion usually occur primarily in the lumen of small intestine, and protein digestion it can continue inside of an enterocyte. Remember, I have the two peptidase, amino and carboxyl peptidase bases, right, can be secreted or can be synthesized in the enterocytes and can continue digest the di or tripeptides inside of enterocytes, right? And carbohydrate and lipids usually finish digestion in the small intestine. This is one point that different between this digestion process of macronutrients. And lipid digestion usually, again, complete in the small intestine. But once it's absorbed, the absorption requires, the vesicle system, right, with the assistance, the bile acid. And it's also something happening in the enterocytes, that means the transport and reacidification process, right, will occur in the enterocytes. And also transport is to the body system is different. Like carbohydrate, we have a glucose transporter system on the membrane, right, of enterocytes, and transport glucose directly to the portal vein. And amino acids also have a transport system, and transport amino acids also directly to the portal vein. But the lipids, we need a lipoprotein, right, to carry the triglyceride, and they also not directly to the portal vein, but first to the lymphatic system and then the circulation portal vein, right? All right. Any questions about digestion, absorption part? Yeah. Oh, yeah, sorry. Endoplasmic reticulum, is the organelle by the cells, ER. Still remember, like mitochondria, like ER, Golgi, have organelle. So ER is a place that synthesizes lipids, right? Okay, now let's talk about carbohydrate metabolism. Oh, sorry. Okay, so 1 and 2, basically we'll talk about general concept of glucose metabolism, and particularly focusing on the glycolysis and TCA cycles. Do you guys have an updated version of notes? Okay. So general concept of glucose metabolism. So how glucose is metabolized in the body through what type of metabolic pathways, right? So we're going to talk about metabolic pathways of glucose metabolism. And again, the metabolism, we need to thinking about the different physiological condition, the glucose metabolized differently through the different metabolic pathway, right? And then we're also going to talk about the glucose homeostasis. That means glucose, particularly blood glucose level, need to be maintained at the normal range. So how our body does something to maintain the blood glucose level at normal range, right? And we also going to talk about the glucose transporter in different tissues. Again, we already know, right? Glucose, cannot be just freely enter the cells and need a transport system to help transport glucose into the cells. And different tissues have a different type of transporter. And this transport of glucose in different tissue also be regulated, depend on, again, the physiological condition, right? And lastly, glucose can be metabolized in different tissue and differently, right? Or in other words, that different tissue metabolize differently, glucose differently. Again, this all related to the physiological condition, whether our body is under the fat condition, eating, or under the fasting condition, not eating. So our body metabolizes glucose differently, all right? So this kind of general concept we need to know. Okay, now let's just go over the overview of the glucose metabolism. So we talk about digestion, absorption, transport of carbohydrate, right? Do you still remember what are the final products of digestion, carbohydrate digestion? Glucose, fructose, and galactose, right? So again, I don't know why. Oh, for some reason. I used the wrong version, but anyway. So, final products of digestion, right? The glucose, fructose, and galactose, right? And cellular fuels, that means, remember this class we're going to talk about energy metabolism, right? So, the nutrients that we absorbed from diet can be used for produce ATP energy, right? So cellular fuels, that means the molecules or nutrients that can be uptake into the cell and metabolized or oxidized and to produce ATPs. So what type of nutrients or metabolites that our body or cells can use for ATP production? I list all these nutrients and metabolites here, right? Glucose, of course, right? And fatty acids, amino acids. These are the three major nutrients, right, that we get from the diet. So we can use these three major nutrients to produce ATPs. But also ketone bodies, have you heard about, right? Ketone bodies, and also the gluconeogenic precursors like glycerol, lactate, and propionate. And this that I highlight in blue, these molecules or compounds that actually were mainly produced or synthesized in our body, right? So our body mainly just obtain the glucose, fatty acids, amino acids from the diet, and the blue highlighted compounds, molecules that our body synthesizes. And these compounds can also be metabolized and used for energy or ATP production, right? But primarily under the fasting condition, when we don't have food, right? When we don't eat food, then we need energy, so our body can synthesize these molecules, for energy production. And next, the glucose is the primary metabolic fuel in humans. That means our the 32 to 70% of energy that our use actually is from glucose, right? So glucose is very important, most important energy fuel for our bodies. So without the glucose, or I think we have a problem, particularly for the brain, right? We we cannot survive if we have a very low glucose level. I'll tell you the reason why later, right? And other point that all tissues are able to use glucose as energy fuel. That means any type of cells in our body can take up glucose and oxidize it and produce ATP. And again, the last point is very important to keep in mind in the class, that physiological state determine glucose metabolic fate, not just glucose but also fatty acids and protein, right? So that means in the fat or fasting condition, glucose can be metabolized through the different metabolic pathways. I just take an example. For example, during the fat condition, when we just eat a meal, and most of the glucose, a lot of glucose, let's say, right, be taken up by the liver and to synthesize glycogen and stored energy, right? Of course, the part of the glucose being directly taken into the cell and for ATP production. But during the fasting, we don't have the glucose available, right? Our body. So our body will break down glycogen from the liver and release glucose into the circulation to raise the blood glucose level. And this glucose can be uptaken by primarily brain, right, for the energy use. So the metabolic pathway is different. And the fat condition, liver does glycogen synthesis, right, pathway, and synthesize glycogen from glucose. And during fasting, and liver actually break down glycogen and release glucose for energy use in other tissue. So again, keep in mind, the fat and fasting condition, glucose can be metabolized differently through the different metabolic pathway, and also different tissues that involve. Okay, now how your body metabolizes carbohydrate. Again, we learn this metabolism at the different levels. First, at the tissue and organ levels. Again, I mentioned different tissues metabolize glucose differently, right? For example, gluconeogenesis. Have you heard about gluconeogenesis? The gluconeogenesis means that the body can synthesize non-sugar precursors to glucose, right? And this gluconeogenesis pathway being more active, particularly during the fasting condition. And only two tissues can do gluconeogenesis. Do you guys know which two tissues? Liver, right? Liver is the most important tissue, can do gluconeogenesis, and kidney. And not the other tissues, like a muscle, adipose tissue, were not able to do gluconeogenesis. So again, different tissue, that's different function in terms of metabolism of glucose, right? And secondly, at the subcellular levels, so inside of cell, let's look at inside of cells. We have a lot of different organelles, right? We have mitochondria, ER, and Golgi, other organelles, and metabolize of nutrients in different organelles, right? In other words, different organelles has a different function in the metabolism, energy metabolism. So in this class, I just wanted to emphasize that mitochondria is the very, very important organelle that does mainly the oxidation metabolism, right? So, glucose, if completely oxidize, you need mitochondria, or fatty acids mainly oxidize in the mitochondria. So mitochondria is very important organelle, does the oxidative metabolism, and also finally to making ATPs, right? ATP is also made in the mitochondria. Right? So mitochondria, again, is very important in the metabolism. And the other important general concept about metabolism is anabolic pathway or anabolic metabolism. I mentioned a little bit, right? So the anabolic pathway means that the synthesis of is the process of the synthesis for large compound molecules from smaller precursors, right? For example, glycogen synthesis. The glycogen is very complex, molecules. So glycogen is synthesized from glucose, right? So this process is considered the anabolic pathway. And catabolic pathway is basically breakdown of larger molecules to smaller molecules, like glycogen breakdown pathway, right? You break down glycogen and these smaller glucose molecules, or triglyceride breakdown pathway, right? You break down free fatty acids and glycerol. So this are the catabolic pathways. And anabolic pathway is a pathway that links between provide kind of link, between the anabolic and catabolic pathway. For example, the TCA cycle, citric acid cycles. Citric acid cycles, a lot of reactions, involve a lot of reactions. So some of the molecules can break down during these cycles, and also this cycle provides some smaller molecule for the synthesis pathway, right? So this TCA cycle is a typical part of anabolic pathway. Right? Any questions so far? No? Okay. Okay, now more concepts. So aerobic pathway versus anaerobic pathway. Do you guys have any idea about these two pathways difference? So aerobic pathway, that means this pathway that requires air or means oxygen, right? So this metabolic pathway requires oxygen. Without oxygen, and this metabolic pathway cannot be occur, right? And here a couple of questions about the aerobic pathway. So where does aerobic metabolism occur in the cells? Any idea? I just talked about a very important organelle. Yeah, mitochondria, right? So mitochondrial oxidation, the oxidative metabolism in the mitochondria requires oxygen, right, or ATP production. And what are the end product of aerobic metabolism? If some molecules like glucose or fatty acids completely oxidize and with oxygen in the mitochondria, what are the final products? Yeah, CO2 and water, right? Okay. And anaerobic pathway. Anaerobic pathway just opposite to aerobic. Do not need oxygen. So without oxygen, our body cells is still be able to metabolize glucose, right? But cannot metabolize fatty acids, right? So without oxygen, glucose can be oxidized, can be metabolized, but fatty acid cannot be metabolized. So aerobic usually occurs in which organelles in the cells, if not in the mitochondria? In the cytosol, right? So in the cytosol, and for example, the glucose can be metabolized through the glycolytic pathway, and this pathway is occurring in the cytosol and can be without oxygen, right? And what is the end product of anaerobic metabolism of a glucose? Have any idea? Lactate, you said? Did you say lactate? Yes. Thank you. Great. So we're going to talk about later, right? The glycolytic product, in the cell that under anaerobic condition, lactate, right? Okay, so very good. Let's see. All right, so what do you need to know about the carbohydrate metabolism? I listed the two slides tables, right? Basically, glucose can be metabolized through 5 different pathways, right? Glycolysis, glycogenesis. Glycogenesis is also called glycogen synthesis, right? And gluconeogenesis, glycogenolysis. Glycogenolysis is a glycogen breakdown pathway, right? And also last one, the pentose phosphate pathway. So we're going to learn these 5 different pathways that glucose can do, and you try to understand whether this pathway belongs to anabolic or a catabolic pathway, or what under which physiological condition, fat or fasting condition, this pathway is being activated or inactivate, something like this, right? And in which tissue does this pathway occur, and what is the function? Why our body need to do glycolysis or glycogenesis? What is function, right? What is the consequences of the activation of this pathway or inactivation of this pathway? And also, you don't need to understand every step of in each metabolic pathway, but you will need to remember the rate-limiting reactions in each pathway, right? And the key enzyme that characterize this rate-limiting reactions, and how is this rate reaction or this pathway activity is regulated by hormones like insulin and glucagon, right? Under the fat and fasting condition, how they are regulated. So this type of the information that you need to learn, right, in this specific session. All right, shall we take a break? Okay, so come back 11:35, right? Okay, so circulating glucose, we know it's very important, right? Has to be within the normal range. So normal range will be, very different depending on the fat or fasting condition. So fat condition, when we eat a meal, that definitely our blood glucose level will raise, will increase, right? And during the fasting, our blood glucose level will goes down, but our body will have, regulated system to not allow the blood glucose level go too high or go too low under, different conditions. So basically we'll maintain, the normal range. For example, after meal, that we will be able to maintain our blood glucose level between 4.5 to 5.5, right? Micromole per liter, or during the fasting, we don't want our blood glucose levels go lower than 3.3. So we'll be able to maintain in the normal, the healthy people, right? Will be able to maintain the normal range of a blood glucose level. Of course, if we eat a carbohydrate diet, blood glucose level can go a little higher than the normal diet. So these are the 5 metabolic pathways of glucose metabolism I mentioned. Oh, yes, question.
What's the difference between the post-absorptive state and the after digestion of carb?
Oh, the post-absorptive phase means just state is the fat state, after a meal. But depend on what type of meal that we eat, right? If we just normal diet, contains a reasonable proportion of carb and lipids and protein, that our blood glucose level will go high to like 4.5 to 5.5. But if we eat a really, high carbohydrate diet, right, then blood glucose level will go higher than compared to people that eat a normal diet, right? And this 5 metabolic pathways. Glycolysis. So glycolysis is a metabolic pathway that converts glucose into pyruvate, right? So glycolysis, glycolytic pathway can occur in both aerobic and anaerobic condition. And again, the products also depend on which condition. For example, under the anaerobic condition, right, you mentioned that the final product of glycolysis should be lactate. But under the aerobic condition, the end product of glycolysis is pyruvate, because pyruvate can be further metabolized in the mitochondria and oxidized to CO2 and water, right? So will not have a lot of lactate generated during the aerobic condition, right? Because again, pyruvate can be further oxidized in the mitochondria. Only when under the anaerobic condition, some problem with the air, with the oxygen supply, or some problem with the mitochondria function, then pyruvate cannot be further oxidized in mitochondria, then will be converted to lactate. So under the anaerobic condition, lactate will be the final product of glycolysis, right? So, and the second pathway is glycogenesis, also called glycogen synthesis. And the third is gluconeogenesis. The gluconeogenesis is a metabolic pathway that can convert the non-sugar precursors. We have multiple non-sugar precursors, including glycerol, lactate, propionate, and some gluconeogenic amino acids, right? All these precursors can be converted to glucose through the gluconeogenic pathway in the liver and also in kidneys. Of course, primarily during the fasting condition, right? And glycogenolysis is also the pathway called glycogen breakdown pathway. That simply, just break down the glycogen and release free glucose. And this pathway is what you expect. Glycogen breakdown pathway should be activated in which physiological conditions? When do we need to break down glycogen? Fasting condition, right? And we need to use the stored energy to break down glycogen and release glucose into circulation. And also I wanted to remind you which other tissues can also store glycogen, a lot of glycogen. Do you remember? Besides liver? Muscle? Yes, skeletal muscle, right? I can also store a lot of glycogen. But why I didn't mention, glycogen breakdown from the muscle during fasting? Yeah. What? Say that again. For itself? Okay. Yeah, I mean, you're right, but from like a mechanism point of view, because muscle lacks of a very important key enzyme called glucose 6-phosphatase, and this enzyme is very important to convert glucose 6-phosphate to free glucose, so then glucose be able to release from muscle to the circulation. Right? Because muscle lacks this enzyme, so muscle cannot release glucose into circulation. Right? So will not be able to directly contribute, glucose to the circulation. Glucose, right? So during the fasting, muscle will not be the primary sources of glucose. Muscle glycogen is not the primary sources of circulating glucose, right? So only when glucose is secreted into the circulation, then can help the other tissues pick up and use for energy, right? If you can secrete it, just only use it for, like you said, for muscle itself, then it cannot help, right? Our body to maintain blood glucose level and glucose utilization in the other tissues. Okay, so the last pathway, we just going to touch a little bit, pentose phosphate pathway. And this pathway will not contribute like, glucose into circulation or glucose homeostasis, but it has its own function. Basically, this pathway, pentose phosphate pathway, is a pathway that synthesizes 5 carbon sugars, right, for like nucleotide synthesis, DNA, RNAs, right? So it is just a different function. Okay. Now let's talk about the glucose homeostasis. The homeostasis means that maintain the blood glucose level at the normal range, right? So how our body will do to maintain blood glucose level under the fat condition or during the fasting, right? We don't want to blood glucose go too high after meal or too low during the fasting. So let's talk about in a fat condition after meal, how our body will do to prevent increased blood glucose level, right? It's lots of tissues involved in this regulation. First, when we eat diet or contain carbohydrate, of course, right? And small intestine digests carbohydrate and absorb the carbohydrate into the portal vein. And some of the glucose used by small intestine. So small intestine, remember I think I mentioned the 2 or 3 amino acids, glutamine, and aspartate, right? And these 3 amino acids from diet is important sources of energy. And in addition to these amino acids, and glucose is also the another important sources of energy for small intestine. So small intestine can have glucose from the diet for energy, and the rest of the glucose being transported into portal vein and to the liver. And in the liver, liver can do actually 5 metabolic pathway of glucose metabolism, glycolysis, glycogenesis or glycogen synthesis, and glycogen breakdown, and gluconeogenesis, right? But not all these pathways are active. Again, depend on the physiologic condition, right? Under the fat condition, the glycolysis or glycogen synthesis pathway are very active, right? Because glucose need to be utilized for energy by the glycolytic pathway, right? And also glucose need to be synthesized glycogen stored in the liver. So these 2 pathways are very active and who can metabolize glucose from the diet. And the glycogen breakdown and gluconeogenesis, what do you expect will be under the fat condition? The activity should be high or low under fat condition. Low, right? Because we already have a lot of glucose from the diet. We don't need to break down glycogen. We need to synthesize and not break down glycogen. And also we don't need to synthesize glucose, right? We have a lot of glucose, so these 2 pathways should be low. And also some of the glucose can be converted to fat and stored in the body. And this is something we need to know, kind of interaction between the nutrient, right? Different nutrient metabolic pathway. So unuptaken or non-metabolized glucose then will release into the circulation. So glucose in the circulation after meal should be increased, right? So blood circulating glucose level increase after meal, and this glucose we can see majority of the circulating glucose, like 22% of the circulating glucose, goes to brain for energy use in the brain, and basically mostly to the glycolytic pathway. And also lots of glucose goes to the skeletal muscle and adipose tissue. And these 2 tissues, the glucose uptake by these 2 tissues is regulated. Remember, we talk about a glucose transporter, right? So different tissues have different transporters that we talk about later. So these 2 tissues, the main transporter, the type of transporter is GLUT4, glucose transporter 4, right? And GLUT4 is the insulin-dependent glucose transporter. That means the activity of GLUT4 is regulated by insulin. When insulin levels increase, so insulin can increase the GLUT4 activity, so allow the GLUT4 to transport more glucose from circulation into the muscle and adipose tissue, right? And of course, the other tissues can also pick up glucose from the circulation. So glucose transport activity is very important, particularly these two tissues. Actually, almost 80% of insulin-stimulated glucose transport occur in the muscle and the adipose tissue. So during the fat condition, the muscle and adipose tissue play a very, very important role in glucose uptake or the reduced blood glucose level, right? So if something wrong with this transport activity, then blood glucose level will not be reduced after meal. So that is the kind of condition related to disease. Do you guys know what type of disease? Diabetes, right? Actually, type 2 diabetes. And also, the pancreas, right? Particularly beta cells in the pancreas as the main cell in the body that releases insulin. So pancreas is respond to the high blood glucose level. In other words, that high blood glucose level can stimulate insulin release from beta cell. So after meal, two things happen. One is the blood glucose level increase, and the second thing is the insulin levels increase, right? So insulin increase then can stimulate glucose to uptake into particularly these two tissues, muscle and adipose tissue, and then result in the decreased blood glucose level. Right? So insulin is very important, and the transport activity of the glucose is also very important. And this, defects in these two activities can result in the diabetes, type 1 and type 2. So type 1 basically is the problems with the insulin secretion, right? The beta cell or pancreas were not able to secrete insulin, so then insulin level will not increase after meal, so then a lot of glucose cannot be uptake into muscle and adipose tissue, and then raise the blood glucose level. This is type 1. And type 2 basically is because of the problem with the glucose transport. Insulin action is reduced. You have we're going to talk about later more specifically type 2 diabetes. But again, some problem with the transport activity, right, of glucose. All right, so this is how our body maintain blood glucose level after meal. Now, how about during the fasting condition? So during the fasting, how our body cannot, to maintain blood glucose, not a lot of glucose, blood glucose level drop, right? This is very important. So we don't have a food, right? So we don't have a glucose from the diet. But our body is able to produce glucose, and particularly from these two organs, the liver and the kidney that are involved in the regulation of blood glucose during the fasting. Liver stored lots of glycogen, right? And during the fat condition, we get a lot of glucose from diet and synthesized, converted to glycogen and stored. And during fasting, the glycogen can be break down and released into circulation, so it can raise the blood glucose level, right? And also, liver and both liver and kidney can synthesize glucose from non-sugar precursors and also release into the circulation to raise the blood glucose level. Right? So during the fasting, liver can do both glycogen breakdown and gluconeogenesis, and kidney can do the gluconeogenesis, right, to make blood glucose level high, to maintain a high level, right? As I mentioned, that glucose uptake or transport activity by tissues is a key regulating mechanism or step that control the blood glucose level or glucose homeostasis. So we wanted to know some of the key transporters and how they regulate blood glucose level, and how they respond to glucose or respond to hormone insulin, right? There are actually 5 transporters that have been well studied, well known. Of course, still people discover the other type of transporters, but we're just focusing on this, and this type of transporters express at different tissues and regulate glucose transporters through the different mechanism. And in general, we just wanted you to remember there are 2 categories of transporter. We divided 5 transport into 2 categories, right? One is called insulin independent glucose transporter, and the other one insulin dependent glucose transporter. So very easy to remember. Insulin dependent glucose transporter, group 4, only one, right? Group 4 is insulin dependent, and the other type is insulin independent, right? So again, I described that dependent means that this transport activity is regulated by insulin, right? Only when insulin level is high and this group 4 activity is high, so it's dependent on the presence of insulin, right? And this table shows us the 5 different types of transporters, and they are tissue distribution, which tissue has this type of the transporters, and proposed function, right? And I want you just to focus on the 2, 4, 5, and SGLT1. And these 2 we already know, right? In the small intestine, remember enterocyte, the SGLT1, right, transport glucose or galactose, and group 5 is also highly in small intestine, right? And transport basically is the fructose, still remember. And group 2 and group 4, they also have a different tissue distribution. Like group 2 basically is high in the liver and pancreas beta cell, and also kidney and small intestine. And group 4 mainly in the skeletal muscle, cardiac muscle, and adipose tissue, right? So how glucose transporter regulate glucose metabolism? So in the fat condition, the 2 major, the main glucose involved in the glucose metabolism are group 2 and group 4. Again, I talked about earlier that insulin-dependent glucose uptake by group 4, right? So about 80% of the blood glucose can be transported into skeletal muscle by group 4, and 20% of blood glucose is transported by adipose tissue during the fat condition after meal, right? When this, insulin level is high. And insulin independent glucose uptake by GLU2 is also very important. Remember, liver and pancreas beta cell have higher GLU2 activities, right? Now this is a diagram and show us more clearly, how GLU2 regulates glucose metabolism in the fat condition, right? So I mentioned after meal, blood glucose levels increase and insulin levels also increase, right? So first of all, when we eat meal, and our blood glucose level increase, and this will stimulate activity in two main tissues, liver and pancreas. Let's look at the pancreas first. Pancreas is a glucose sensor, mean when glucose level is high in the blood, and GLU2 will transport the blood glucose level in the beta cell and triggers the insulin secretion, right? So again, blood glucose level is a major trigger of insulin release from the beta cell. So this something happened in the pancreas, and the liver also has a GLU2 and transport glucose into the liver cells. And liver can do glycogen synthesis, right? To convert the glucose to glycogen and store in the liver. And also, glucose can be metabolized and converted to fatty acids, right? So, liver under the fat condition can serve as the energy storage organ, right? To store, glucose as glycogen or converted glucose to the fatty acids. So all this activity in these two tissues can lead to decrease the blood glucose level, right? And I just want to mention further that insulin, the consequences of insulin increase or leads to uptake of glucose into muscle adipose tissue, right? So this all, tissue plus the muscle and adipose tissue together, these four tissues, organs work together to reduce the blood glucose level, right? And then, so here I wanted to go a little bit detailed about insulin secretion by high levels of glucose, stimulated by high levels of glucose. Try to remember this diagram, how glucose triggers insulin release, right? Mechanistically. So pancreas has both beta cell, it's a major cell that make insulin and release insulin, and alpha cell and produce the glucagon. And this is the main type of transporter GLU2, right? So when glucose level in the circulation is increased after meal in the fat condition, glucose uptake by the GLU2 into the beta cell, right? This represents the beta cell. And glucose being metabolized through the glycolysis pathway and produces the ATP. So ATP and ADP ratio is increased, and this will lead to the potassium, the depolarization of cell membrane, and the potassium channel is open, right? So you have efflux of potassium, and in the meantime, you open the calcium channel, and so calcium will enter the cells and leads to increase the concentration of calcium inside of cell. So concentration of calcium inside of cells is a very important trigger of the insulin secretion, right? So it can stimulate the movement of insulin secretory granule and to the cell surface and release insulin, right? So several steps. Group 4, uptake glucose, and glucose metabolized by glycolysis and increase ATP to ADP ratio and causing the depolarization of cell membrane, right? And potassium channel open, an efflux of potassium and influx of the calcium, and increased calcium concentration inside of cells, and leads to the insulin secretion, right? So you have the two questions: which tissue organ produces insulin? Beta cell, right? Pancreas. And why insulin is produced? Why we need to produce insulin? What insulin does? Why we need insulin? To allow the glucose uptake from circulation into which tissue? Mainly adipose tissue and skeletal muscle, right? And so we need insulin to push more glucose enter the primary skeletal muscle and adipose tissue, and this is the mechanism that can reduce the blood glucose level, right? Okay, now let's see. So I just wanted to mention that this process called the first tier response to elevate the blood glucose level. That glucose stimulate insulin release from beta cell. This process is called first tier response to elevate the blood glucose level. All right. Okay, let's see. I think I'll just stop here, right? And then we have, maybe I can finish actually this slide. So I just mentioned the first tier response, right, to elevate blood glucose level is the glucose stimulate insulin release. And now we talk about a second tier response to elevate blood glucose level. So once insulin is released, and what happened next is the insulin will regulate or stimulate glucose uptake from circulation into the skeletal muscle and the adipose tissue, right? So this insulin stimulates glucose uptake into the skeletal muscle and adipose tissue, and this process called second tier response to blood glucose level, right? Does that make sense? So and so I just put all this together. So the after meal elevated blood glucose level triggers first tier response, which is the insulin release from beta cell, right? And then once the insulin levels increase, the insulin can start second tier response, which is stimulate glucose uptake by skeletal muscle and adipose tissue. So all these two response, and involve the multiple tissues, liver, the pancreas, and skeletal muscle and adipose tissue together. They all work together to reduce blood glucose level after meal, right? So now we have a couple of fill in blank. So glucose transporter and tissues are involved in second-tier response in glucose metabolism. So which glucose transporter involved second-tier response? Group 4, yes. And which tissues that involve second-tier response? Skeletal muscle and adipose tissue, right? Yes. Oops, sorry. I don't know why this doesn't work. Well, do you guys remember, right? The transporter is group 4, and tissue, skeletal muscle, and adipose tissue, right? Okay. And what about the first-tier response? Which transporters that involve first-tier response? Group 2, and which tissues involve first-tier response? Which tissue? Pancreas, right? Beta cell. Okay. All right, so we have informal writing. Okay, so try to describe how blood glucose levels are maintained after meal and fat state and during fasting. Right?